Good morning, everyone. My name is Adriana Serna, and I am the Communications Director for the Caribbean Hotel and Tourism Association. I'd like to take the time to thank you for joining us today for the CAS Sustainability webinar on the value and benefit of coral reefs. Now I'd like to introduce you to Ms. Danae Hines. She is the co-chairperson for CAST and the Director of Sustainability for OBM International. Danae?
Thank you, Adriana, and thank you everyone for joining our webinar. We're really excited, and I would like to thank Dr. Dahlgren, who is our speaker today, who has taken time to give us this valuable information on this interesting topic. Just a little bit about Dr. Dahlgren. He is a marine ecologist who studies a wide range of topics related to tropical and marine ecosystems. He has extensive work, including studies of mangroves and coral reef ecosystems, marine protected areas, and populations of dynamic fishery species ranging from Caribbean fisheries, such as the Nassau grouper, Caribbean spiny lobster, emerging to fisheries like sea cucumbers and parrotfish. He's done amazing work that has led to dozens of peer-reviewed publications and has influenced marine resource management and policy, not only in the Bahamas, but also through the Mesoamerican reef and throughout the wider Caribbean region. His current work now focuses on improving the management and expanding the network of marine protected areas in the Bahamas and elsewhere. And what we're really excited about is the fact that he has worked to do some restoration of coral reef and mangrove ecosystems throughout the Bahamas and other places. So he's going to share with us some of the very important benefits of our coral reef systems and also maybe some key insights into just coral reef management and restoration. So without further ado, I will hand it over to Dr. Dahlgren. Thank you so much for being here, and we appreciate this information. We're looking forward to it.
Thanks a lot, Danae. Can everybody see my screen? Just want to make sure.
Yes, we can.
Okay, yeah. Perfect. Yeah, so like Danae said, have been very much a generalist in marine ecology throughout my field, but really the common theme throughout all my work has been looking at ways to improve stewardship of marine resources to the benefit of marine ecosystems in general and the people that depend on these ecosystems. And recently that has really become a little more focused on marine protected areas, on the conservation side of things, and also looking at restoration on the other side. So what I'm going to talk about today with respect to coral reefs and climate change is really looking at what we can do to, a little bit about what we can do to prevent coral reefs from declining, but also looking at what we can do to reverse that decline through restoration and other techniques. So just in the course of the webinar today, I'll cover a little bit of the value and importance of coral reef ecosystems and how climate change impacts coral reefs, and then what actions we can take in light of that decline to help coral reefs recover from these impacts. And I'll get into a little bit of some of the lessons we've learned about reef restoration from around the Caribbean and pulling on some specific details of some of the work I've done in the Bahamas. So just to really start with the basics, what is a coral reef? And the top bullet point here is one I pulled from a textbook, I think. It's a wave-resistant limestone structure built dominantly by the vertical accumulation of coral skeletons. So basically what that means is it's whether it's a shallow rocky area where the waves are breaking or a deeper area, it's been rock created by living coral over decades, centuries, millennia. And the types of reef really vary a lot. Like you can see in the pictures here, there's different species of corals that have different growth forms. Even the same species of species of coral might have a different growth form based on the conditions that it lives in. So the depth of a reef, wind and waves, light, temperature, amount of sediments in the water all affect how corals grow and how that reef forms. So we have an incredible diversity of reef types in the Caribbean, from barrier reefs and atolls to much deeper reefs and everything in between. But they have in common that they were all built by living corals, and the structure that the living coral makes provides habitat to fish and other species that rely on that reef for shelter and for food. And I promise I won't get into too much biology, but just this is important when we start talking about how climate change affects corals and what we can do to restore them, just understanding a little bit about coral biology. And basically, one of the first questions is it an animal, is it a plant, or is it a mineral? Well, it's all three. The coral polyps, the little tiny things that look like little sea anemones that have tentacles and a mouth, are animals. When they grow, they reproduce asexually by dividing and forming. One colony will divide and form two, and that'll continue so on, to form a large colony of coral. So that's the animal part. That polyp can feed on things floating around in the water column with those tentacles, but it does rely a lot on microscopic single-cell algae called zooxanthellae that live inside the animal tissue, inside that coral tissue, which are little plants that provide a lot of nutrients to the coral, a lot of food to the coral. So it's a symbiotic relationship. The zooxanthellae, the little microscopic algae, are getting a nice sheltered place with a lot of nutrients, and the coral in turn is getting a lot of energy out of the plant there. And that's really important in terms of having corals, allowing them to grow and reproduce and produce the limestone skeleton, the rock, that actually builds the reef. So when we look at a coral, the outer layer is a mucousy layer. That's the animal. Inside it, the thing that gives it its color, otherwise it would be transparent, is the plant part. And the rocky structure that it forms is the limestone rock that the coral forms to build the reef. So all this is very important because as the corals are affected by climate change and other impacts, it affects both the plant and animal part of it. And for us to restore the reefs, we need to know a little bit about this biology. So getting into then, why do we care about coral reefs? Why are they important? From an ecosystem point of view, they're extremely important. They're one of the oldest ecosystems on the planet, and they really are, they are often compared to being rainforests of the sea, which is a really accurate comparison. In some metrics, they can exceed rainforests in the diversity of life that lives on them. They provide food and shelter to fish, invertebrates, and other organisms. Most, or 25% of all marine fish species live on coral reefs, even though coral reefs are only 1% of the Earth's surface. Globally, 4,000 species of fish and 800 types of corals are found on reefs. In the Caribbean, we are not as species rich as the Pacific and Red Sea areas, but we do have between 500 and 700 species of fish that are found on coral reefs, and 50 to 70 types of corals typically found on the Caribbean reef. But getting beyond the biological importance, they're also extremely important to people. Approximately 850 million people globally live within 100 kilometers of coral reefs. That's a huge amount of the population of the planet that lives in coral reef areas, watersheds that affect coral reefs. Coral reefs are an extremely important source of food globally. They protect shorelines from wave damage, storm surges. They're the first line of defense, really, against storm surge in most areas where they're found. And then we're also just beginning to discover how this diversity of life that we find on coral reefs translates into having chemical properties that can be used to develop drugs, whether it's antibiotics or anti-cancer treatments. We're starting to discover how coral reefs might advance medical science tremendously. So they're extremely important in a number of ways to people, not the least of which is economics. The value of coral reefs globally is about $10 trillion, it's estimated, with about net benefits per year of about $30 billion. And that can be divided into important things like tourism and recreation, which are important to all of you, coastal protection, fisheries, and also the diversity of life conservation aspect. So coral reefs are an important economic driver in places where they occur and are significant in the global economy as well. But we're seeing that coral reefs are rapidly declining in their health. Going from the pictures on top, an elkhorn coral reef and patch reef area with high amounts of living coral to areas where there's not much living coral is left. The skeletons are being covered with algae and being eroded away by sponges and other things. And what we're seeing is as the coral goes, so goes the diversity of life and the amount of ecosystem services, the benefits that they provide to people. And we've really seen this over the past 30 to 40 years in particular. I promise these are only 2 slides I have that have graphs on them, but basically the take-home message from this one is that a number of studies over the past 10 to 15 years have shown this decline of the percent cover of living coral on a reef, the amount of a reef, when you look at it, that's alive across the Caribbean has gone from anywhere from 40 to 50% live in the 1970s down to 10% live around most of the Caribbean at present. So in my lifetime, we've seen that decline from 50% to less than 10%. So that's a very rapid decline, very sudden, and there's a number of reasons why we have that decline. But one of the significant things about that decline too is that as coral goes away, seaweeds are rapidly taking over reefs, and this is being described by scientists as a phase shift. We're going from a coral-dominated ecosystem to a seaweed-dominated ecosystem, a macroalgae-dominated ecosystem. And this graph is just showing that in the top left of it, in the 70s to 80s, when we had high coral cover on a reef, we had low cover of seaweeds, macroalgae, and that's changed. It's flip-flopped over the past 30 to 40 years. And what we're seeing with that change is that the amount of reef structure has declined, the number of species has declined, the abundance of individual species have declined, and we're losing a lot of what we once had as coral reefs. As far as the causes of this decline, we really can look at it at multiple spatial scales. At the global level, we see climate change as being a huge driver in coral reef decline, particularly elevated sea surface temperatures, but also things like increased frequency and intensity hurricanes. Ocean acidification is another thing. I won't get into too much details about that, but it also has helped drive the decline of reefs in general. On a regional level across the Caribbean, some of the same factors like increased sea temperatures, have driven decline, but also we see things like regional disease outbreaks, invasive species, most notably recently lionfish, and the die-off of key grazing species that kept that macroalgae in check in the past. Things like the long spine sea urchin diadema have died off and haven't recovered in many places. So a lot of these sources of decline are ones that are removing live coral from the reef, killing the coral itself. Other ones, like the grazer die-off, are preventing the reef from being able to recover from these other impacts. So we both lose live coral on the reef, and also the resilience of reef are decreased by these things that cause declines. On a more local island level, and this is just a Google Earth image from Bahamas, including New Providence and Andros Island, but could be applied anywhere in the region, really. We see some of the same things impacting reefs. Elevated temperatures can vary from one island to the next. Disease outbreaks can vary from one island to the next. But we also see things like inappropriate coastal development, developing in sensitive areas that's impacting reefs, and overfishing some key species that help keep the balance on reefs as well. And then if we drill down further to a more local level, at the individual reef level, we see a lot of the same factors, plus you can add in hurricane damage or damage from vessel groundings or anchors or divers or fishing gear. A lot of these local impacts accumulate over time and cause significant declines in coral reef health in general, both, again, in terms of the amount of live coral on a reef and in terms of other key species that help keep the balance on reefs functioning so that the coral reefs can recover from other impacts. So for the next little bit, I'm going to focus really on the climate change impacts, and I'll just address the hurricane one first. A lot of the climate change models are showing that with increased temperatures in our climate, we're likely to see increases in hurricane intensity and frequency. So the damage from individual hurricanes are going to be more intense, and this photograph is off of Nassau last year after Hurricane Matthew came through, and we saw huge coral heads the size of a dining room table being flipped over and knocking over every other coral around them. So huge sections of reef were knocked down like bowling pins. Now, hurricanes have always been around. Reefs have survived for centuries of hurricanes. But because we see their ability to recover decreased from things like overfishing or the loss of the long spine sea urchin, they're not able to recover quickly. Plus, if they're getting hit more and more frequently by hurricanes, they don't have time to recover before increased damage occurs. So reefs are just not able to recover from this. Some will recover faster than others. All reefs are a little different, but in general, we're seeing that reefs are not getting enough time to recover from these impacts. But hurricanes can be really locally hit or miss. After Hurricane Matthew in the Bahamas, we saw some areas that were devastated, like in this picture. Other areas hardly saw any damage at all. So a more pervasive issue related to climate change that is occurring on a global level, with some regions around the world more impacted than others, is coral bleaching. Now, the coral bleaching is when, remember the biology lesson earlier, the microscopic zooxanthellae, the little plants that live inside the coral tissue, give the coral its color. Well, when corals are stressed, temperatures are too warm, temperatures are too cold, too salty or too fresh water, they get stressed, and one of their first things they do is they spit out their zooxanthellae, leaving just the clear animal tissue that lets you see right through to the bare limestone rock. So it appears like it's been bleached. That's what I'm referring to when I talk about coral bleaching. So when this happens, the coral is losing that zooxanthellae, that algae that provides it with a lot of nutrition, a lot of food. So one of the first things that happens is the coral will still be alive, but won't be able to grow as much because it's not getting enough food, and it won't be able to reproduce as much because it's not getting enough food. So the coral is basically still hanging on, but it's not reproducing and growing. And this might last for a period of time after this bleaching, after the coral regains some of that zooxanthellae. In severe cases, when bleaching lasts for a long period of time because temperatures are elevated for a long period of time, or the temperatures have spiked in a severe way, the corals will die. And we have seen mass bleaching events more and more frequently since the 1980s, with some global events occurring, or at least regional events, in 1998, where in some reefs in the Bahamas, off of Andros, for example, the Andros Barrier Reef lost 50% of its corals in a single year. And in other parts of the Caribbean, 2005 was a devastating year for corals. And over the past 2 years, we've seen major bleaching events occur as well, with significant amounts of coral dying globally and specifically in parts of the Caribbean. So this really is one of the biggest impacts of climate change, and a lot of these events have been associated with El Nino years, but we're seeing with climate change that in general, sea surface temperatures are increasing, and we're seeing these events occur more and more frequently. So this is a major threat. It is important to realize, though, that not all corals or not all zooxanthellae are going to respond in the same way. So some might escape bleaching based on some of the environmental conditions of where they live, and some might be more tolerant of changes in sea temperature, increases in sea temperature that occur during most of these bleaching events. And that's important when we start talking about restoration. So what can we do then in light of these changes? Well, the first thing, probably the most precautionary thing we can do, is to reduce the number of local threats to keep reefs resilient. We can't do much to change climate change in a short period of time, in a period of time necessary to help reefs. But we can do things like protect important grazers like parrotfish, avoid building in sensitive areas, or adopt building practices that reduce runoff or sewage leakage onto reefs, create marine protected areas, and to educate people about how their actions affect coral reefs. We can do all of those things that will reduce some of these local stresses that compound these global impacts and help build resilience or maintain resilience of reefs. So we need to do that. In addition, we can start actively restoring reefs, and that's what I'm going to focus on for the rest of the time here. Coral restoration. Really, I don't like the restoration is the common word used for this. I don't like that. It's really, we're not able to restore things to where they once were at this point, but we can help them rehabilitate. We can give them a chance to improve their condition and really jumpstart a natural process that helps corals come back. When I first started in my career, never even considered this as a possibility. Corals grew so slow, we wouldn't be able to do this, was the general consensus. But over the past 10, maybe 15 years, techniques have been developed that really make restoration a realistic possibility, at least on a local level. And there's three main types of restoration activities. And I'll talk about each one, the in situ, on-site midwater nurseries, land-based microfragmentation, and sexual reproduction. So to go through what I mean by each of those, this is the in situ nurseries. These midwater nurseries are something that many of you might have seen. It's becoming more and more common, probably the most common technique for restoration of corals, particularly in the Caribbean. Whether it's a tree nursery like in this picture or a line nursery, which is like a clothesline with corals hanging from them. It's been proven effective for some of the more endangered species of corals, the elkhorn and staghorn corals, particularly staghorn coral in the Caribbean. And the advantage of doing this is we can take a small cutting of coral that's an inch or two in size, 5 centimeters or less, hang it from a line of these nurseries, and within a year we can get significant growth, up to 30 centimeters or more, much faster growth than the way they naturally grow when attached to the bottom, because we can grow them in ideal conditions, and by hanging them in the water they're getting a lot of flow across them, which brings them more nutrients than they would on the bottom, that sort of thing. We can do this for a few species, and we can produce in an individual tree like this is typically 50 corals can be grown there. So we can produce hundreds or thousands of corals annually to return to the reef. And I'll talk a little bit more about the pros and cons of different approaches later. But this is one that's tried and true, and we've really got this down to a decent science. The second technique is called microfragmentation. It's more recently developed and requires a land-based facility at this point to do. What we do is we take, it's based on the same principle that corals naturally reproduce, pieces break off, they reattach to the bottom and can grow. Under natural conditions, they are usually overgrown with algae, that sort of thing. But for the branching corals, if we put them in a line nursery or a tree nursery, they don't get overgrown as much because they're off the bottom. Or if we take them into a lab, we can cut them into tiny pieces of about a centimeter or so of just the living coral tissue and a thin layer of skeleton underneath it, and glue those to a little substrate, a little disc or tile. And when corals are broken into those small pieces and given the ideal conditions for growth, we can grow them about 10 times faster than they grow naturally. So you can see the progression of two different coral species, one on the top, one on the bottom in the diagram. We have 1 centimeter or so little pieces of coral placed a couple centimeters apart, and we can see that by 140 days they're starting to fuse back together again. For natural growth to occur, that would probably take a couple years for that to happen. So in 139 days we can get a couple years of growth. We can do this with a number of different species. The midwater nurseries, the trees and the lines, we typically do with branching species that are fast growing, the elkhorn and staghorn corals. With this, we can do with just about any species of coral, including some of the slower growing mounding corals, the mountainous star corals, the brain corals, things like that. So we can produce hundreds of colonies that are reproductive size from fragments taken from naturally occurring ones in a fairly short period of time. Within a year or so, we can definitely get them to the size where we can start putting them out on a reef, and they'll be reproductive adults at that point in time. The third technique is a departure from these other two. These other two are based on taking a cutting from a coral and letting that cutting grow into a new colony, or in the case of microfragmentation, fusing into a new colony. What we have in this third approach is based on sexual reproduction, based on collecting the gametes that the corals produce. And this is a close-up view of an elkhorn coral, and you can see it releasing bundles of gametes, eggs and sperm together in each of these bundles. And the coral will produce millions of gametes. And we can collect those by putting a tent-like net over a coral. These gametes float up, and we can collect them in a tube at the peak of that tent. Take them back into the lab, fertilize the eggs. For many corals, you need to, they won't self-fertilize. Eggs and sperm are produced by a single colony, so they have both male and female reproduction, but they won't reproduce with themselves, so you have to cross them with other colonies. We can do that in a lab. We can grow the larvae, and in about a week or two, we can get them to settle onto specially designed tiles. And after a couple months, put those tiles out on the reef and let those corals grow. So from the millions of eggs that we can collect in a single night of spawning, we can produce tens of thousands of colonies for outplanting potentially, but at much smaller sizes than the other techniques. So then the question is, which approach is best? Well, there's pros and cons to each one. Probably the lowest cost, least amount of technical expertise would be the in situ nurseries. The problem is that it's only good for a few species. The yields are in the hundreds versus tens of thousands potentially. And because you're taking cuttings from corals, fragments from corals, you're limited to the genetic diversity that you can collect versus on the other end of things, the sexual reproduction, you can mass produce corals. You can produce tens of thousands that'll have high genetic diversity because you're doing crosses of gametes there. The problem with that is that the corals that you have to outplant are at a much smaller size, so they're at greater risk of being killed by a predator or overgrown with algae, that sort of thing. So there's not really a one-size-fits-all approach. It really depends on the species that needs to be restored, the scale or the scope of the restoration, and the status of the reef that you're restoring. Is it an area of very low genetic diversity, high genetic diversity? Are you really just supplementing with some new corals here and there, or are you looking to regrow the whole reef? All of this varies, and really a combination of approaches eventually is, I think, what we're going to see more and more of. But for right now, really the in situ nurseries is something that's taken hold and really a proven technology that can be done at low cost, sort of a backyard nursery sort of thing, if you will. There are some important factors to keep in mind, though, doing restoration. That the restoration itself doesn't remove the threats to corals. So if there is an ongoing threat, sedimentation or sewage outfall or damage from anchors, if there is ongoing threat like that, the corals that you put out in a restoration are not likely to have a great chance of surviving. So you need to deal with the threats in addition to restoring reefs. The second is that restoration is really limited in its spatial scale. We can restore a hectare of reef, an acre of reef, not hundreds of acres at this point in time. So really what we're doing when we're restoring a reef is either bringing back a couple species that are totally absent or very rare at a site. Like in this picture, all the elkhorn coral around is dead except for this one colony. Well, that one colony isn't doing a lot of good to help repopulate the reef if it can't reproduce with another one. So if we can get a few other elkhorn colonies growing around this one here and reproducing, we have a chance of jump-starting that reproductive process that can help replenish that reef and some of the surrounding ones. So really we have to be realistic about the scale of what we're doing. It really is helping jumpstart some natural processes. There are other aspects of reef rehabilitation to keep in mind. And this particularly related to climate change, this first point. Some corals may be genetically suited to survive bleaching events. They may be more thermal tolerant, or they may have zooxanthellae living in them that are more thermally tolerant. They'll be able to tolerate higher temperatures. And we can selectively use those in our restoration attempts. We can select those corals that are more likely to survive climate change scenarios, or even get ones that are more resistant to disease outbreaks. So we can really be selective in what we do and screen for those corals. The second thing is that we really can target areas that are important for replenishing areas around them. What we've done in the Bahamas is really looked at picking which areas are going to be the most productive for reseeding surrounding areas and actively target those for restoration. We've been working with oceanographers and geneticists to see how populations are linked, to see where the source populations are that are going to replenish surrounding areas. The third point there, site selection and management of sites is very important. And I'll get into this in a minute or two with the Bahamas example. We see that the corals that we produce in nurseries and outplant to reefs are more likely to survive under some circumstances than others. Small differences in temperature, small differences in fish communities, small differences in predators on corals may have a great impact on the success of the restoration. And then finally, it's not just restoring corals. We need to keep in mind we need to restore the resilience of reefs as well. So doing things like bringing back the long spine sea urchin, doing restoration of that species, or protecting parrotfish or other key species is important to do hand in hand with the coral restoration to make the restoration most effective. To get to the example from the Bahamas, and this is using the Bahamas as an example because I've been most involved and most experienced there, but we could also choose examples from Florida Keys and some other places. We're really, over the past 10 years, we've learned a lot of lessons about reef restoration or coral restoration and reef rehabilitation. In the Bahamas, we're establishing this reef rescue network where there's more than a dozen sites in the Bahamas right now with some form of coral nursery and coral restoration activities. And we're trying to bring everyone together to learn from each other's experiences and to provide some scientific, some technical expertise to some of the practitioners, which are dive shops and resorts, and some local conservation organizations, provide them with the technical expertise to really be more effective at what they're doing. So in the Bahamas, the first nurseries we established were back in 2011 for staghorn and elkhorn corals. This picture shows a line nursery design, but around the same time there were some other nurseries established using the coral tree design in 2012 and 2013. In this case, we chose to use a line nursery where the corals are suspended from these horizontal lines because it's an extremely low maintenance type of nursery, and we were only able to get out to the site twice a year to maintain it. And each line nursery will produce 200 to 300 coral fragments for outplanting annually. And so just to show you a little bit about what that growth looks like, in 2011 we started with pieces of corals that were about 3 to 4 centimeters in size. And this is with, example shows staghorn coral. I'll show elkhorn coral in a minute. So that was the start in May of 2011. By May of 2012, this is the same piece of coral against a 1 centimeter grid background on an 8.5 by 11 inch piece of paper. So you can see that in total it went from being about 4 centimeters or so to about 30 centimeters in length and had branches going off in different directions. What we did was we cut off the new growth, outplanted that to the reef. We got 2 outplants. One on each end of the initial nub, and we took that initial nub in 2013 in the middle picture on the left, So I'm sorry, that was May 2012 is that picture on the left. Let it grow for 2013, and the next year we had even more growth on that, producing 5 outplants of corals, each about 15 centimeters or 6 inches long. We cut that new growth off, went to the bottom left picture there, which shows the nub at its starting point of growth for the 2014 growth year, and we see even more growth, enough to produce 7 outplants in that 6-inch or 15 centimeter size range. And then in 2015 we did the same thing, and this is what we got in 2015, and at that point we stopped measuring because we would just get so much growth. And I believe in 2016 we cut this off the nursery entirely, outplanted the whole thing, and started fresh. So in a 5-year period, a 4-year period, we got about 30 pieces of coral outplanted from that initial 4-centimeter piece. And we did this 250 times over throughout the line nursery. For elkhorn coral, it's a similar story. We started with slightly bigger pieces of coral. You can see 1 year of growth. It's growing 2 arms, one at either side. We cut off that larger arm, outplanted that, and started the growth process over again. And the following year, 1 outplant. So it's a much lower yield, but still a consistent yield of about 1 outplant in that 15 centimeter or 6-inch size range each year from elkhorn coral. When the coral gets outplanted, we attach it to the substrate with a 2-part underwater epoxy, put a tag on it so we can track its growth rate over each time. And this is a piece from that same coral I showed you in the nursery. That coral started out, year 1 is the upper left picture, year 2 is the upper right picture, year 3 the lower left, year 4 the lower right. And this is after 5 years. So that same piece of coral went from a 6-inch, 15 centimeter outplant. Part of it was covered with epoxy to attach it to the reef, and has grown into something that's probably about 50 centimeters across and 25 to 30 centimeters high, little patch. And we have, after doing this for 4 or 5 years, over 1,000 corals outplanted on this one reef, both elkhorn and staghorn. And this picture just shows you the same progression for a piece of elkhorn coral from that same one that I showed you in the nursery. This is the first branch that was cut off in the upper left, and it's grown over a 4-year period into year 5 here in this picture. And by year 4 or 5, it reached the size where it is reproducing. And we have a number of corals in close proximity to each other, so we're hoping that there is some reproductive success, some egg fertilization going on. We haven't measured it yet. Hopefully next year we will get to measure that. But we didn't have 100% success in the Bahamas. In this particular study, we did it on 5 reefs in the southern Abaco area, 2 main reefs. And what we have found was on one reef we would have 90% survival of corals from 1 year to the next. On the other reef, it was only about 60% or so. And the reason for that is that on the reef that had lower survival, we had these little snails that are voracious predators on this species, of the 2 species of corals. It's a little snail called Corallophilia breviata, and that really was limiting our success. So we've had to go back and see if we can manually remove these snails, and I also have a student working on what natural controls of these snails might be. But had we known this ahead of time, we could have really screened and selected our reefs a little better where there weren't these predators. In other parts of the Caribbean, the fireworms, bristleworms are voracious predators on staghorn coral, so those are something to look out for as well. So there are different factors that make some reefs more suitable naturally than other reefs, and we're starting to figure that out. So site selection is key. and also monitoring growth and survival so you can see how effective your restoration is. So just starting to wrap up here, some of the take-home messages, really, the important things to remember are that the climate change may be the biggest global or regional threat to corals and species that live on coral reefs, that depend on coral reefs for habitat and food. We can help the recovery process. The recovery process is damaged. The reefs are not resilient because of factors like overfishing, die-off of the long-spine sea urchin. But we can help recovery in some areas, in areas where other threats are minimal. There is no one-size-fits-all approach. There are several techniques available depending on what the goals are, what the budget is, what species of corals are present or not present, what the environmental conditions are. So we have options now. Some are more advanced than others. Others are a little bit experimental still, but within a year or two, even those, I think, will get to the point where they can be done mass-producing corals easily by non-technical people. And then finally, to reiterate, other threats to corals have to be reduced for the restoration to be effective. As far as getting started, because there's so many options, there's a number of different starting points, but really I would just say some things to keep in mind if this restoration is something that you would want to get into or support. The first, site selection is critical, both selecting sites for setting up nurseries, selecting sites for the actual restoration activities, and even selecting where your corals that are going to go into the nurseries are from, all play a role in determining success. So really getting off on the right foot with a good foundation is extremely important here. The second thing to keep in mind is what the initial investment is. I would recommend to anybody to start small and then scale up after a year or so. The different techniques that I went over briefly all have different costs associated with them. The different nursery types for the in situ midwater nurseries, whether it's a tree nursery made out of PVC or the line nursery, all play a role. But a pilot nursery project, material costs, you can probably do for definitely less than $1,000 to get something set up and going. I know costs of materials are going to vary from place to place, so I don't want to narrow that number down, but that's the level of investment roughly that it would take to get something meaningful started. But more importantly is the time and time resource commitment. All the different techniques I went into require some amount of maintenance to nurseries, or for growing corals if it's a lab-based facility. No, there's a typo here. Outplanting corals also takes some time on an annual basis. Some of the PVC tree nurseries that I've established require maintenance at least on a monthly basis. The line nursery that I showed the picture of, we only had to go out to twice a year for effective growth. If we were able to get out more, it probably would be even more effective. But it still was about 90% survival of corals with only getting out twice a year. So the initial startup cost of the line nursery was probably a little more than tree nursery though. So there's a little bit of trade-off there. But really I would budget time to be in the water for maintaining nurseries, checking on things, making sure there's not a disease outbreak or the corals aren't subject to some bleaching event or something if the site selection wasn't the exact right conditions for them. And then outplanting corals, it does take time to harvest corals from the nursery, whether you're cutting the whole coral off or cutting off a fragment like we were doing in the Bahamas, and epoxying those or cementing those or attaching them to the reef in some way take some time to do. It'll take a couple days for that, for several people. Another important consideration is just monitoring the success of the things. You don't want to invest all of this effort and then just let it go out there and not know if those corals are surviving and growing. You really want to track the success. If things are not working out well, learn from the lessons, try to figure out why they're not doing well and not make the same mistakes again. And being able to show what successes you do have. And that gets into the final thing, sharing experience. There's a lot of experience out there, particularly with these in situ midwater nurseries, particularly the tree nurseries. So there's a lot of little tips and tricks that people have developed that help success or reduce cost. There's also becoming a more of a network approach to these things. In the Bahamas, we've started to establish this reef rescue network. On a more broad scientific and government level, there's a coral restoration consortium that has been established following a meeting in November with a bunch of scientists and practitioners. So there's a lot more sharing of information right now, learning from successes and really, fine-tuning things a little bit. And some of these are available online as resources. There's a couple of online resources that I've listed here. The Reef Rehabilitation Manual and Coral Reef Restoration Handbook they're a little bit dated now, to be honest with you. This has been such a rapidly advancing science in the past 5 to 10 years that things that even came out 4 or 5 years ago are already probably out of date. Still provide good rules of thumb and some best practices, but we are rapidly moving beyond them. So they're a good foundation maybe, but maybe not the Bible to rely on for moving forward with these things. A lot of more up-to-date information is available on this Reef Resilience Network, which is something that the Nature Conservancy, I believe, has set up. And they have a lot of current information on that website that is worth checking out. On that same website is the home of the Coral Restoration Consortium that I mentioned. This is really a consortium that does involve some practitioners of reef restoration, but primarily scientists. It's a consortium largely funded by NOAA and the Nature Conservancy, and is trying to really work on advancing reef restoration science and provide more information to people that are interested in getting involved in reef restoration. And then for some of the different techniques, there are some organizations that have a lot of expertise. For the in situ tree nurseries, the Coral Restoration Foundation, based in the Florida Keys, developed the tree nurseries. They've grow thousands of corals using them each year. They have recently expanded internationally around the Caribbean, so they are a good resource when it comes to that particular type of nursery. Microfragmentation is a technique largely developed by the Mote Marine Lab in Florida, in the Florida Keys, their center there. But others in Hawaii are doing it, and there's a for-profit company called Coral Vita that's setting up shop, possibly in the Bahamas, possibly in the British Virgin Islands that'll be doing it. So that's an area that's going to get more and more attention and more resources out there. For sexual reproduction, SeaCorps International has been one of the pioneers in dealing with that. SeaCorps International is an organization composed primarily of public aquaria. They've been doing a lot of research, particularly in Curacao and Mexico over the years. And then the organization I work for, the Perry Institute for Marine Science, has been doing a little bit of each of these, and I'm always willing to help and have staff that can help as well. So I will end there. Thank you for your attention, and I guess we'll open up for questions now.
All right, thank you. Okay, so we have, the good thing is that we have one of the questions that we started out with is if a resort wants to start coral rehab, what's a good resource to get them started, and kind of a step-by-step guide. That was a question from Sarah Burnett, and you basically answered that question in this last section. So there you go, Sarah. I hope that answers your questions. If you have any further detailed questions for Craig, you can go ahead and contact him. Let's leave your contact information up there so people can see it. And let's see, what other question we have? Is tourist sunscreen as much of an issue for coral restoration as previously reporting has led us to believe?
That's a really good question. I believe in some cases it can be. In places where there's a lot of water flushing, current flow, it's not as much an issue. In a big open bay, that kind of thing. If it's an enclosed area, it can be an issue. That's for sure. It is probably a bigger issue for some restoration techniques than others, maybe sexual reproduction, because those larvae and newly settled corals are so sensitive. It's probably more of an issue than with some of the bigger fragmentation sorts of approaches, the in situ nurseries or microfragmentation.
Okay, another question is there any evidence that coral reefs are moving deeper due to warm oceans, sort of like how some trees are moving up mountains? Or is it all just happening, too fast?
It's really happening too fast. We do see with some species that the shallow water ones are not the reproductive populations anymore, that it's only the deeper ones that are responsible for reproduction and repopulating shallow areas. But there's not a lot of evidence that the deeper ones that corals are moving deeper or even moving north or south based on how waters are warming. It's just happening too fast for that to really occur.
Okay, and Sarah, again, she wants to know where are you, in the Bahamas are you currently working?
The short answer is all over. We have projects, whether they're coral nurseries or reef assessments, that I'm working on throughout the northern 2/3 of the Bahamas. So last month I was doing coral spawning collection and raising gametes out of Nassau. Next week I'll be doing something similar off of Eleuthera. I have nurseries set up in Andros and Abaco and other places, Exuma, so really all over.
Wonderful. Well, thank you so much. Any additional questions that we get from participants, please go ahead and submit them, and we'll make sure to give them to our speaker, and he will answer you after the webinar is done. We are currently out of time. Thank you all so much for participating, and thank you so much, Craig, for giving us all of this wonderful information. Until next time, everybody have a great day, and again, thank you so much for your time.
Thank you, everybody.